Um, Welcome to session 2 of the BDBIO Symposium 2025. My name is Nitya. So we continue the previous theme in this session as well, that is precision medicine. So we have a great lineup of speakers today. The first two talks will be for 25 minutes with 10 minutes of Q&A. So our first speaker is Dr. Kishore Jaganathan, who will be joining us online. Kishore is an AI researcher and a director of deep learning at Illumina. The title of his talk is Variant Interpretation of Rare Genetic Disorders Using Deep Learning. Yeah, over to you, Kishore.
Okay, sounds good. Thank you. Hi, my name is Kishore. I'm a deep learning engineer at Illumina. So, Illumina, for those of you who are not familiar, we are one of the main manufacturers of DNA sequencing machines. So, I'm on the computational side. My work is focused on why you should sequence your DNA, what value you can get out of it, and not on the hardware side. So, today I'll be presenting. Giving you an overview of some of the projects that me and my colleagues have been working on for over the last 8 years. So in the title, like, so the three keywords here: variant interpretation, rare genetic disorders, and deep learning. So I'll give you a quick one-slide overview of each one of them, so everyone is on board. Um, I will. Yeah, so are my slides moving forward, I hope. Yeah. So I'll start by giving you an introduction of what rare genetic disorders are. It's defined different ways in different countries, but a general definition is if it affects less than about 200,000 people. Some examples include like Tay-Sachs disease, Duchenne muscular dystrophy, and Wilson's disease. So each one of them is completely different from each other. Like Tay-Sachs disease, for example, it affects it's a mutation in the hexa1 gene. And when you have that mutation, like your nerve cells tend to die, and the person doesn't survive beyond a few years. And DMD, it's a mutation on the DMD gene, and so this one's a little less aggressive. Your muscles become weaker, and eventually you again die. Like you only live for more around 20-25 years. Wilson's disease, it's a mutation on ATP7B gene, and this starts like copper starts to accumulate in your body. And this one won't result in death, but like if you manage properly, you can live, but it'll again be a difficult life. So each one of them is very, very different from each other. The actual causal genes are very different. Throughout the world, about 10,000 of these rare disorders are cataloged. And when you collectively look at how many people are actually suffering from these diseases, it's about 350 million. That's almost 1 in 30 people. So while these are called rare diseases, like when you put them all together, it's actually quite common. And the one thing that is common to all of them is many of them are genetic in origin. So for about 80% of them, the typical way the disease happens is because there's one or two mutations that you have in your genome that, depending on whether it's a dominant or recessive mode of inheritance. And each of these diseases, it's often unique to some families or some individuals. And for most of them, very little is known. For only some of them, you even know like which are the genes that are causing the diseases, and because of that, again, most of them don't even have any approved treatments. Again, these ones have not received attention from pharma traditionally, because even if like drug discovery is a very expensive process, and even if you end up finding a drug, it only helps a small fraction of people, and each one of them like is a completely different drug. So it's something that's not been like given a lot of attention to by the traditional modes of drug discovery. But this has been a lot of focus of the precision medicine approaches, because if you know which of the mutations are actually causing diseases, then you can like do things like gene editing or CRISPR or antisense oligonucleotides and fix that. And this one, well, I'm oversimplifying it, like regardless of which gene the mutation is, like okay, if you can CRISPR the same way, then that's one way of like looking at all of this in a unified manner. So a simplistic way of like an oversimplified solution would be like, okay, can you find the mutation, and once you find the mutation, then can you edit it? Is like that would be like a way you can think of how to treat these. From our end, like we are focused on the genome sequencing side, so our work has been to see if how can we find the actual mutation that is causing these diseases. So a typical workflow for a rare disease analysis would be like, okay, the patient will first start sequence their genome, and you can find about 4 to 5 million variants per individual. And out of those 4 to 5 million, maybe one or two of them are actually what you're looking for. So it's a very, very challenging problem. So you start by applying some filters based on common knowledge that you already know. So one of the things that people always look for is if the variant is common in the population, then it's very unlikely to cause a genetic diseases. So there are many population databases like for example Nomad or UK Biobank, like for many European individuals, like these ones collectively, like there's almost about 1 million or 2 million genomes that are available. So what you typically do when you sequence a rare disease individual is go and check if that variant is present in the common population, and if it is present, then it's unlikely to cause diseases. And once you apply that filter, you can bring down the number of variants from 4 to 5 million to 20 to 30,000. And then you split it into whether the variant falls in the protein coding regions or in the non-coding regions. The non-coding regions, our understanding is very, very poor, so we don't even look at those for their disease analysis. And predominantly, it's focused on the protein coding side. And even over there, you mostly look for what are called protein truncating variants. So these are variants that cause frame shift or stop gain. And very likely, these are going to create a faulty protein. So these ones are most likely going to be damaging. And sometimes you also look into missense mutations if you have an additional line of evidence, like if it's a de novo mutation or if it's in a highly conserved position. Once you do all of this, like you can bring it down to a few hundred variants. And then there are again more databases where it tells you for different genes, like which are the kind of diseases they cause, like OMIM or ClinVal, ClinGen, PanelApp, and so on. So here you can think of it as there's a list of genes and like which are the diseases they cause. Now, depending on whatever disease the individual has, like if the variant falls in a relevant gene, then you can consider it as likely causal. And the pipeline like this typically today like has about 20% successful diagnosis, and a lot of the missed opportunities because yeah, you're throwing out a lot of missense variants, and also more importantly, like the entire non-coding genome, 98% of them is completely thrown out because we don't fully understand them. What we have been looking into over the last few years is like, can we use deep learning to solve this particular problem? So the main problem that we're trying to solve here is you have a variant, and now you want to predict whether that variant is going to be harmful to you or harmless to you. And how do you represent the variant for this prediction problem? So you cannot just say chromosome this position, this is the variant, and because that's something the deep learning models don't understand. So there are a few different ways that tends to work out better is so there's three different options like that seems to work out well. So one of them would be you provide two sequences as inputs. One would be you have the position of the variant and the actual genomic sequence. You can choose whatever window you want depending on the problem, and one sequence with the variant, one sequence without the variant. That will be your input, and the output will be whether the variant is going to be harmful or harmless. Or you can do another way where you build a deep learning model where it will only take one sequence as input and gives you some sort of prediction for that sequence. You can, for now, think of it as a fitness score. So whenever you have a variant, you can try the reference sequence and get a reference fitness, and you can try the sequence with the variant and get an alternate fitness. And if the fitness changes too much, then you can flag that variant as something that is harmful. And if the fitness doesn't change too much, then it is harmless. And lastly, this is more inspired by all of the language models that have been becoming very popular in the recent years, where you can pretty much take any things like text and the internet, and then just create lots of fill in the blanks problems where you just hide random words and ask the neural network to predict what words actually like were hidden there. And even a problem like that, people have shown that you can actually understand a lot of grammar and so on. And there is analogies to it in genomics as well. We can have an amino acid sequence or a protein sequence, and you can set up prediction problems where you just hide a position and ask it to predict a nucleotide. What typically happens, the hypothesis here is if a nucleotide is safe, most likely it is going to predict it because it's okay. And if a nucleotide is dangerous, it's not going to predict it because it's not seen in the healthy genome. While the premise is very fair one, there are lots of challenges that are unique to genomics, because when you look at all the fields where deep learning has had the most amount of success, like there's a few things that are true over there that's not quite true in genomics. So one of them is so when you look at fields like text or video or audio, like the general population is very, very good at understanding that mode of data. So you can collect a lot of high quality training data by just crowdsourcing the efforts. Like if you like just want to like know what object is present in an image, those are all tasks that the general population can solve. But for things like these, like I think you need like a more scientific background, and even then it's a very challenging problem. So in terms of the amount of training data available, it's very, very less. And also secondly, when it comes to more popular fields like in deep learning, like there are very, very solid benchmarks that are like curated by a large number of people. So if you're today working on like image processing or text processing, there's already like 30 or 40 benchmarks that you have ready access to, and that is something that is not there in this particular field as well. And without benchmarks, it's very difficult to tell whether a new model that you train is it better than the one that you already had. And even over here, I mentioned three different styles, which one is better, and things like that. So I will give you a quick overview of three different models that we have trained. So I'll start with one for missense variants. So over here, the goal is you have a missense variant as an input, and you want to predict whether that variant is harmful to you or not harmful to you. In terms of the actual label data that is out there, the closest one that we have is websites like ClinVar and HGMD, and these are again scientific. You can think of these as scientist crowdsourced in the sense that people have curated. Anytime people write a paper, and if they have some variants that they have discovered that are dangerous, they actually go and report that in this particular website. It's like actually built over like contributions from hundreds of people, and like the final list of variants is about a few thousand, which is nice, but again, like nowhere close to what is needed to train these kinds of deep learning models. So we have to try doing something more creative. So what we do over here again. So I mentioned previously that yeah, variants that are common in the population are unlikely to cause rare diseases. So when you like go and look through all these like huge population databases that are out there, you can find about 125,000 variants that are actually common in the missense variants that are actually common in the population. So now you immediately have like a data set of 125,000 variants that are like missense variants that are harmless. And again, the problem with this approach is it has already hit saturation. You can like now sequence many more human beings, and you still end up keeping on finding the same mutations and again because like when you look at the history of human beings, like there's a huge bottleneck that happened a long time ago, because of which we all share too much of the genome. So you can't really find many more common variants. What we have done through many bioinformatic analysis is found that variants that are common in the primate species are also actually benign in human beings. And this is something that is not immediately obvious, but it's been validated multiple ways that the statement is actually true. So what we have done over the last few years is we have worked with primatologists across the world, and even from India, including from CCMB. We've sequenced 809 primate individuals spanning 233 species. And from them, we have identified 4.4 million variants that are common in the primate populations. And again, the main reason why you see so many common variants in primates is because, again, this spans a lot of different species, and they don't have some of the founder effects that we humans have. So it's already like 70x bigger than anything else like that we previously had. So all of this solves the problem of having a really large, high quality, harmless missense variants. What about harmful, right? It's harmful one, like you cannot really, there's really no way around it. But what you can do is through statistical analysis, again, it's well known that 1 in 2 missense mutations are actually harmful. And there's a total of about 70 million missense mutations you can find. So when you remove these 4.4 million, like you're left with roughly 65 million missense mutations. Of these, you know, roughly half of them are harmful, but you don't know which ones are harmful. But the really nice thing about deep learning is they're very, very capable of tolerating a lot of label noise. Like, so even a 50% label noise doesn't really stop deep learning from learning well. People have shown you can even push it to 80, 90% label noise. It's perfectly fine. And so what we do now is set up a benign versus pathogenic prediction where you have 4.4 million benign variants, which are completely pure. And pathogenic, out of the 65 million, we choose a 4.4 million that is completely matched to the benign set in terms of like what reference amino acid, what alternate amino acid is. That part is only 50% pure, but that's something that's perfectly fine. We know the models can handle. And in terms of the neural network that we train here, it takes it tries to solve three different tasks, and at the input level it takes three different inputs. One, for whenever you have a variant, we provide the amino acid sequence around that variant, like about 100 amino acids on either side of the variant. And we also provide sequence conservation information through multiple species alignment. What does that position look like in many other species? And we also provide the protein structure. These are AlphaFold predictions for that particular protein as additional input as well. And the reason why we provide the protein folding information is we have found, looking through many examples from ClinVar, that, for example, in the protein you're seeing over here, all the blue circles you see are where the benign variants lie, and all the red circles you see are where the pathogenic mutations lie. And typically, where pathogenic mutations lie, there are things like domains, and it's a very structured region. And where benign mutations are like it's mostly very unstructured. So structure is a very useful feature to add as input as well. So we have three inputs like amino acid sequence, conservation, and protein structure, and the output will be yeah, we predict whether the given variant is going to be benign or pathogenic. And on top of it, we also try to, taking inspiration from the large language models, also try to solve some fill in the blanks problems where we hide some amino acids and ask the model to predict it. Also hide parts of the structure and ask the model to predict it. And again, all of these choices, the way we decided on them is through creating extensive benchmarks. So for each problem, when we start, the first thing we do is through extensive bioinformatic analysis, try to set up many benchmarks, and then all decisions that are made are based on which ones give you the best results on the benchmark. So over here for the coding side, we have 6 different benchmarks. So the very first one, so what's called assays. So these are like typically MPRA for so it's like you can just create a synthetic mini gene and over there at the same time in a single experiment measure the effects of hundreds or often thousands of variants. And people have done this for few tens of proteins and for all missense mutations in there, just see in the synthetic mini gene like what kind of effects they have. So you can now see whether the model's predictions are how correlated are they to the experimental effects you made. And the other ones, UK Biobank, it's a massive project by the Europe, like UK government, where they've sequenced about 500,000 individuals and also have extensive health records on top of the whole genome sequencing. So we try to predict all the health records that they have from the whole genome sequencing data and try to get a metric out of that. And lastly, I also mentioned the ClinVar database, where it's a database where there's a few tens of thousands of variants that have annotations for whether they are already known to be benign or pathogenic. And lastly, there are also many uh, sequencing efforts that have like very pure data sets which you can make benchmarks out of. So these are like for different diseases where what they have done is they do like sequence 4 people from the same family. Usually it's mother, father, and 2 siblings, and only one of the siblings have a disease. And why that helps is because you have the parents sequencing as well, you know exactly which ones are the de novo mutations for both the kids. And because one kid has a disease and other doesn't, you now have a very clean control set and a very clean case set. And now you can see whether the models consistently assign high scores to the sibling with the disease as opposed to a sibling without the disease. And so these ones are available for a few diseases, and like we've used all of that and processed them into benchmarks that we can use to evaluate whether the model is good or not. And typically the way a pipeline, the way it works is like every time you start a project, you just create 5 or 6 benchmarks like this, and all choices like what's the best way to model them is all decided by which ones provide you the highest accuracy over here. And this one's on the coding side, but on the non-coding side, things are even more harder in the sense that so all the main premise that helped us actually make progress in the coding side is that 1 in 2 missense mutations are actually harmful, which means that we can have a pathogenic set with 50% purity. Like over here in the non-coding side, is most mutations are harmless, which means there's simply no way for us to get a reliable harmful data set. So if I just pick a random mutation as harmful, I'll be right like maybe 0.1% of the time or 0.2% of the time. That's just way too much noise. So there's just simply no way to get the harmful like a list of harmful variants. So we have to work around that problem. So the way we work around it is so many of these non-coding mutations, they end up causing diseases simply because they end up breaking something inside the cell. For example, a mutation in the non-coding genome end up changing the way a gene splices, or it ends up changing the way the amount of expression like a gene has, or it changes the way a gene gets polyadenylated. And if any of that happens, because of that is the reason why the variant actually ends up causing the disease. And here, suppose we have a model where it's a perfect predictor of splicing. So if I give you a sequence, it tells you exactly how it splices. Then what I can do is whenever I have a variant, I can provide a sequence with the variant and without the variant and try to get splicing predictions. And if the predictions remain the same, then that variant does not change splicing. And if the predictions actually change, then that variant changes splicing. Now I can flag it as, okay, variant that changes splicing. And the nice thing about framing it this way is we can build, there's a path to building good predictors for each of these mechanisms I mentioned here, simply because through a lot of high throughput sequencing, like assays like RNA-seq, or you can easily find out, know, like how exactly like genes splice or how much a gene expresses, and things like histone modifications, where do proteins bind, which part of the genomes are accessible, not, and even for poly, for everything, there's lots of assays which we can use to model these different parts of the cell, like cellular machineries, like through which these non-coding mutations actually cause diseases. So for splicing, what we do, yeah, so we make use of RNA-seq. So over here, so there's one particular data set called GTEx, which is very, very handy, because even when you look at RNA-seq, a lot of the data sets that you see online usually come from blood. And that's not, again, the reason is because it's the one that's most easily accessible. You just have to do a blood draw and then do a sequencing. And but the problem is, in blood, only about 3 to 4K genes are reliably expressed, which means like we don't know how the rest of the genes splice. Like the thing about this GTEx, it's Genotype Tissue Expression Project, is it's about 800 individuals who volunteered that after they die, essentially their body can be used for any scientific research. And for 53 tissues were harvested, and like lots of sequencing was done on all of those. So this one, for about 800 individuals, you have like 53 tissues worth of RNA-seq covering the entire body, which means any gene you pick, you end up finding at least a few tissues where it is expressed. So we make use of that, and to try to build a splicing predictor here. So the model is essentially takes 10,000 nucleotides as input, and for each position, it tries to predict whether it's going to be used as a splice site or not. And the way we use it to predict variant effects is, yeah, whenever you have a variant, you provide the sequence with the variant, without the variant, and check whether splicing changes. If it changes, then yeah, it will be called a like splice altering variant. Otherwise, like not. And simply in terms of like benchmarking, like you can um, uh, so given that like most positions in the genome don't splice, you can't have simple metrics like accuracy. So you have to come up with something that takes into account the class imbalance. So you do use area under the precision recall curve, and also one other metric inspired from protein contact maps, like top K accuracy. Like you see that I think through the non-deep learning solutions are so far behind, and deep learning is one of those things that really helps you solve this particular problem. And so I mostly speak about variant effect prediction here, but there's other things that you can use these sequence to function models as well. Like, well, you can think of it as like you can perform many computational experiments using it. So you can just take a random region and just simply start mutating each position to see like how the model responds. Over here is one particular locus where you just mutate different parts of the genome, and here you see that when like in human introns, almost all introns start with a GT and end with an AG. So whenever I mutate that position, if you see a larger height, that means the model tells you splicing changes too much. And there's this part which is called the polypyrimidine tract, which is again very important for splicing. And here, this one's the branch point, and this one is an SR binding protein. So all these factors that are important for splicing, you can just recover computationally. In this particular case, splicing is a very well-studied problem, so none of these biological findings are new. But if you do have a problem where it's not so well biologically studied, like these kind of tools can be used to at least generate hypotheses for what are going to be the influential factors. And another one, which is our most recent work, is we did something similar for gene expression, where so the goal here is to try and find variants that non-coding variants that cause diseases through changing the amount of expression of a particular gene. At least one thing that was nice in splicing is that each gene provides you about 10 to 15 splice sites, which means like from a human genome, like you have about 300,000 splice sites. But for gene expression, each gene only provides you one value. So like it's a lot less data compared to what we had for splicing. So we had to leverage more deep learning tricks. So over here, we take advantage of something called multitask learning, where when you're trying to, when you have multiple different learning problems and they're all related, instead of modeling them separately, you can try to model them jointly. So in this case, we try to model gene expression alongside things like histone modification, DNA accessibility, TF binding, and so on. And the reason, again, why we chose this is there's lots of high quality data available through consortium databases like ENCODE and Roadmap, where through across hundreds of cell types, we have uniformly processed data for all of these assays. So we train a big model where the input is a sequence about 20,000 nucleotides, and just try to predict all of these quantities. And as a second step, again, so these days a lot of deep learning models like also do a fine tuning step to improve accuracy. So over here, through GTEx, where there's lots of whole genome sequencing and RNA sequencing data. Like we have curated a list of 5,000 variants that are known to alter gene expression. So we use that to fine tune the model's predictions as well. So over here, again, like before, we have about 6 different benchmarks. So 3 of them are again MPRA based. So this is through a small mini gene reporter. You just insert thousands to 10,000s of variants and try to measure their expression effects, spanning promoters, enhancers, and EQTLs as well. And there's many other things you can do here. So there's also very well-known EQTL. So you try to find whether the model can classify EQTLs that cause overexpression, EQTLs that cause underexpression, or EQTLs that have no effects. And there's also protein level measurements for many individuals in the UK Biobank project. So we use that to create another benchmark as well. So there's a total of 6 benchmarks which we use to make all modeling decisions here. And I'm showing you one example here. So this is a famous cancer example where so there's a G like binding protein like in this particular locus on the TERT promoter. So there's multiple of them. Anytime a variant hits there, people have like studied these variants and shown that it's very well known to cause cancer. And when you have a model like this, you can just go and mutate every position to just see how much the model says that the expression changes. So what I'm showing here is the nucleotide height is proportional to how much the model thinks the expression changes. You can see that whenever you're inside that GA binding protein like motif, like the model assigns a very high score, and as soon as you step outside of that, it doesn't assign a high score, which means the model knows exactly, okay, there's a GA binding protein and that has to be intact, and anytime something bad happens over there, gene expression goes up. And so these are different ways to build confidence for, like the models actually doing the right things. And again, going back to the big picture, like what we were doing. So in these rare disease settings, like we have tried running the pipelines without any of these AI methods and adding all of the 3 different methods that I described now, one for missense, one for splicing, and one for gene expression. A combination of these 3 AI methods with that, you can pretty much double the throughput. So that's the main takeaway from all of these 3 different projects that we have. So to summarize all of the work, so deep learning can accurately predict the effects of variants. We mainly focus on applications in rare disease, but there's also applications in other things like fine mapping or things like drug target discovery and so on. There are some specific challenges to applying deep learning in this field because, again, like these are all, there's not enough high quality data set to begin with, or also, like there's not enough benchmarks to begin with, like compared to some of the other fields where deep learning has done well. But there are options. Like in this particular, usually we rely on large scale public databases. Like for when you look at AlphaFold, it had used PDB, and in our case, like we use a lot of ENCODE, Roadmap, and GTEx. And to evaluate our models, it's either purely whole genome sequencing, or oftentimes you do 2 things for the same set of individuals. One of them will be whole genome sequencing, and the second one will be either RNA sequencing or protein level sequencing, or some synthetic mini gene reporters, EQTLs, GWAS, and so on. And what I just presented is what we've been doing from the past, but what's ongoing work. I think on the protein side, there's a lot of focus on large language models. They do seem to do really, really well. So the inputs are a combination of sequence, protein structure, and multiple sequence alignment. Just build large language models on them, and then eventually try to fine tune them on curated lists of variants for which we know what their consequences. On the non-coding side, the large language models don't really seem to be doing that well. Like they seem to be quite terrible. Like I think in the intergenic regions, this kind of fill in the blanks problems like doesn't really seem to work out well. So our main focus on building like really large sequence to function models where the input will be a sequence and the output will be pretty much anything you can measure through high throughput sequencing. And once you're done with that, then you do a fine tuning step on curated data sets for any particular problem that we are looking to solve. So that's all I had in terms of slides. So if there are any questions, I'd be happy to take.
Yeah, thank you, Kishore. So if you have any questions.
Kishore, thank you. Can you hear me?
A little bit, yes.
Okay. Can you hear me now?
Yeah.
Okay. So one of the challenges of rare diseases that anyone faces is actually penetrance. Now, I wanted to know in your model, could you show the predictability across multiple penetrance type?
So like these are more general purpose problems, general purpose models. So I think like all of that will become uh, for some of them it will be downstream and for some of them not. Like things like splicing or gene expression, we don't really tell whether the variant will cause a disease or if it does, like how serious it will be. What we the only thing we can tell with these two models, the sequence to function, the splicing or gene expression, is whether it will change splicing or not, and for what fraction of the transcripts. And now, as a post-processing step, then you have to decide, like for each gene, how much gene expression can be tolerated, how much splicing change can be tolerated, and so that will be like a next step. So it's not included. In the other case, things like missense variants, like where we already have benign and pathogenic. Because the labels are quantized as 0 and 1, I think it will be a bit challenging, but I would not be surprised if the model learns that a little bit. But usually we think of that as a post-processing step where, yeah, this one's come one step before and afterwards, like you then have to decide based on that gene, based on each gene, you have to appropriately decide whether, um, uh, yeah, like that kind of change will be tolerated or not.
Okay, thank you.
Hi, Kishore. Wonderful talk, and congratulations. I think your paper just came out this morning. So I had a question about promoter AI. I haven't actually had chance to read through it, but in terms of being able to sort of draw conclusions across phylogenetic tree, are you able to sort of take a model trained on one species and
The question is not audible at all. So you have to come
uh,
to the other mic which the host was using. I think that one's the cleanest. Yeah.
okay, should I? Can I try again? Is this better?
I can repeat it if.
Yeah,
Yeah.
this one is the cleanest. Yeah.
Okay, my question is about, can you use promoter AI which is trained on one organism, and if you project it onto other organism, does it perform as well?
So his question is, if you develop a model for one organism and you project it on another organism, does it work as well as
Uh,
it must?
it it has to be. My preference is that someone at least does basic amount of testing. Um, like what we have seen is that it does generalize well, and most likely it will be your best bet. That does not mean that you cannot do better. Like I think your best bet will still be if you have models that you can train, you can use principles that we have used here and train a model for the other species. That would be the best bet. But what we have seen in the past, again, is that these things do generalize well to at least some species, like land species, like not things that fly and not things that swim, but things like primates or even like yeah, dog and cow and everything. Like it does seem to. We have done some basic amount of testing, so we focus on the other way. We try to use their genomes to help human prediction because that's kind of what we are most interested in. But that also tells us that it can work the other way as well. But today, if you ask me, I will say as long as it's a land-based and land-based mammal, it will be your best bet. But it will be safer if you can do some basic amount of testing. And if you have the data, I would say even better thing to do would be to just use the same principles. Just a lot of the training pipelines are publicly available. You just yeah, replace the training data with whatever is available for that species, and you should be able to get an even better one. Yeah.
Hi, Kishore. Can you hear me? Okay, fine.
I will try, but if not, the other person has
Okay,
to.
yeah, sure. So,
Yeah.
thank you so much for the talk. I actually have a poster on this exact topics, but focusing on neuromuscular disorders and the pathogenic prediction. So, yeah, my question is about: are you trying to, bring in explainability in why certain predictions are being performed? Like, what I want to ask is, typically a clinician would do an ACMG type of classification. So, what is the kind of reasoning that the deep learning model is doing? Are there efforts in that direction to bring explainability into these models?
So his question is whether you're trying to bring in explainability to the predictions.
We do some stuff. Like I mean, uh, it's not a complete black box, but at the same time, it's not completely explainable either. Some of the basic, like there are, I think at least in biology, like I think there's two things that we can do. One of them is yeah, anytime you're interested in any position, you just one by one mutate every position in that locus, and you end up creating these the kind of plots I showed, where you can see motifs pop up very, very nicely. And if the right kind of motifs pop up, and if the score becomes zero right outside of that, that builds confidence. And another thing you can do is try to treat the deep learning model as a in silico experiment setup. Like if you know like a certain feature is useful through experiments, then you can try and see if you can reproduce that through deep learning. So like, for example, in the splicing case, we know through experiments that there are certain exon lengths, certain intron lengths, nucleosome positioning, and everything that's actually important for the splicing process. And we were able to reproduce all of that through in silico experiments, where we just pick random exons and then just keep changing the exon lengths or intron lengths. And when you're hitting those optimums, you see best predictions. And as soon as you move away, like the predictions become lower and lower. So there are things you can do when it comes to deep learning. It's very, very well known for performance, but at some, I mean, and it's not a complete black box, but there will be there's some amount of explainability that is like no, not going to happen. The way we sleep well is just make sure that way, like we don't just use one benchmark or two benchmarks, because sometimes like no benchmark is ever perfect, and you might end up like moving in the wrong direction. So usually we rely on 4 or 5 minimum. If you're making progress on all of them, most likely that the chance that you're making progress for the wrong reasons is very, very, very low. So that's the one that like I think really brings us the most amount of comfort. Yeah.
Thanks, Kishore.
Hi Kishore, thanks a lot for a wonderful talk. Is it possible for mRNA genome sequencing and as well as RNA genome sequences variant prediction with the help of splice AA and as well as with deep learning models?
Could you repeat the question? I was sorry.
Yeah, sure. Is it possible with mRNA sequencing as well as RNA sequencing for the variant with the help of splice AA algorithm and as well as with the deep learning technique, can we able to predict the results as well?
So his question is it possible with mRNA as well as
RNA genome sequencing.
RNA genome sequencing to predict?
To predict with the help of deep learning models and with splice AI algorithms as well.
Okay. With the help of splice AI, is it possible to predict with both mRNA as well as RNA?
Genome sequencing.
Okay, genome sequencing and mRNA to use splice AI is his question.
I don't think I fully follow the question. Yeah. Maybe you can email it back to me. Like I'll try to respond. I don't quite follow what the question is. Yeah.
Sure.
Okay, so you can get back to him later. Yeah, thank you very much, Dr. Kishore, for your wonderful talk.